Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 20th November 2020. We are into the revision mode and we are revising chapter number 10, cell cycle and cell division. This chapter is very important as far as the CBSE syllabus is concerned, as well as lot many questions in your NEET as well as UPSC ND exam. So you need to focus on this. So what is basically a cell cycle and how important it is, and what is a cell division? So the sequence of events by which cell duplicates its genome, all the genes that are present, synthesizes the other constituents of the cells. In our previous chapter we talked what are all the organelles that are present in the cell, constituents of the cells, right from cell wall, cell membrane, cytoplasm, mitochondria, Golgi body, ribosomes, all that, and eventually divides into two daughter cells is called cell cycle. So a single cell dividing, forming two or more than two cells, and this happens with a cyclic changes of events. So that cyclic changes of events constitute a cell cycle. So DNA synthesis occurs in one specific stage of cell division, but distribution of chromosome in cells occurs in complex series of events during cell division. So when these daughter cells are formed from the mother cell, or we say parental cell, from parental cell daughter cells are formed. Okay, if two daughter cells are formed from one parental cell with both the daughter cells having same constituents, then we say it is mitosis. On the other hand, from one parental cell, four daughter cells are formed with reduced number of chromosomes, then we say meiosis. Mitosis takes place in all the cells, and we must remember number of chromosomes we normally write 2n. So 2n refers to diploid. Ploidy means a set of chromosomes, and that ploidy is referred to as n. When we write 2n means two sets of chromosomes are present. So in whichever cell two sets of chromosomes are present, that cell is called diploid cell. Like in our case, n is equal to, we have got 23. So since we have got two sets of 23, 2n, that is 46 chromosomes, then all the cells that are having in our body, right from skin cell, muscle cell, bone cell, hair cell, you name the cell, every cell has got two sets of chromosomes diploid, except the gametes. The sperm and the ovum will not be having 46, instead they will have 50% reduction, 23. If there is only one set of chromosomes like the sperm and ovum I said, then we say n, that is haploid, half of the original 2n. So whenever a cell is having n number of chromosomes, one set of chromosomes, like in our case 23, then we say such cell like sperm and ovum will be referred as haploid. So basically we have got in our body two types of cells, diploid cells and haploid cells. So during the cell division, all these things will be decided. So in mitosis cell division, the diploidicity is maintained. The identity will be maintained, identity of the species will be maintained because of the maintenance of 2n, diploidicity. Whereas in meiosis, identity will be lost, the anatomy will be lost, the biochemistry will be lost because of non-maintenance of diploidicity, because diploid cells will become or will produce haploid daughter cells. So during this cell cycle, what are all the phases? Like human cell divides once in approximately 24 hours, which may vary in different organisms. In yeast, it takes about 90 minutes to complete the cell division process. So cell cycle is divided into basically two basic phases. That is interphase. First one is called interphase. It is the phase between two successive M phases, that is mitotic phases. Interphase lasts for 95% of cell cycle. This phase is called resting phase, but during this period the cell prepares itself for nuclear division by cell growth. Of course, I should say that this resting phase term was used previously, but now everybody understood that it is a preparatory stage. Hence, it is very active. In fact, that is before the war breaks out, how soldiers train themselves hard for the possible war. Similarly, when the cell is going to undergo division, the cell prepares itself. What are all the constants required? Required cytoplasm, required proteins, required enzymes, all this will be prepared. So we cannot say that interphase is a resting phase. Instead, the most apt word is preparatory phase. So interphase, I should say, is a preparatory phase. And the second one is the M phase, that is mitotic phase. When the actual cell division or mitosis occurs, it starts with karyokinesis. As all of you know, in Latin, karyon means nucleus. Kinesis means breaking. What is breaking? Nucleus is breaking. Hence, nuclear division. So in any division, the first one that takes place is karyokinesis. And this will be followed by cytokinesis. Cyto means cell. Kinesis breaking. Cell is broken. So first nucleus will be broken, followed by cell breakage. And that completes the mitosis. So duplication of chromosomes and end with cytokinesis or division of cell matrix, cytoplasm division. The interphase basically is divided into three further phases. So as you are seeing in the diagram, you can see here M phase. So it is divided into three phases. That is what we call G1 phase, S phase and G2 phase. So three phases are G1 phase, S phase and G2 phase. And of course the M phase. Right? Let's see the three phases. So G1 phase represents the interval between mitosis and initiation of DNA replication. Cell is continuously active and grows in size. So as we said, interphase has got three subphases. G1 phase, that's gap 1, S phase, S for synthesis, and G2, gap 2 phase. So during synthetic phase, or let us talk about the G1 phase. It is a state of initial growth of newly formed cell, in which the cell carries on active metabolic activities in preparation for DNA replication. This is important. Many times NEET exam as well as UPSC, during which phase DNA replication takes place. So you have to remember G1 phase. In G1 phase, a cell has three options: to continue cell cycle and enter S phase, stop cell cycle and enter G0 phase, that is resting phase, for undergoing differentiation, or get arrested in G1 phase, where it may enter into G0 phase or re-enter cell cycle. So three options are there for the cell. Some cells such as nerve cells and erythrocytes RBC that do not divide further exit G1 phase to enter an inactive stage called G0 phase, or G0 phase is called quiescent phase or G0 phase of the cell cycle. They remain metabolically active but no longer proliferate unless called upon to do so. This is the G1 phase. Then S phase or synthetic phase. So this marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. DNA is increasing. Look at that, DNA gets doubles per cell. If the initial amount of DNA is denoted as 2C, then it increases to 4C. So in the beginning it is 2C, and at the end of S phase it becomes 4C. However, there is no increase in the chromosome number. Of course, chromosome number cannot be disturbed because the organism or the cell loses its identity. In animal cells, during this S phase, DNA replications begins in the nucleus and the centriole duplicates in the cytoplasm. So these are the various things that take place in S phase. Now we go to G2 phase. In G2 phase is a stage of further growth of the cell and its preparation for division. During this stage, synthesis of RNAs, say there DNA, now it is RNAs. Synthesis of RNAs and proteins continue. So again you must remember for your UPSC as well as NEET exam, protein synthesis takes place in G2 phase. This is what you must remember. In adult animals, some cells do not divide or may divide occasionally. The cells do not divide further and exit to G1 phase to enter the quiescent G0 cell cycle. In animals, mitotic division is present in only somatic diploid cells, but in plants it is seen both in haploid as well as diploid cells. Mitosis cell division is also known as equational division because the numbers of chromosomes remain same in parental as well as progeny cells. So in next we need to talk about the M phase, that is when the actual cell division mitosis occurs, it starts with karyokinesis, nuclear division, and followed by, I said, cytokinesis. So M phase is having the characteristic of karyokinesis and cytokinesis. It's the phase of cell division which may occur by mitosis or meiosis. M phase separates the products of chromosome replication and distributes the same equitably into two daughter cells. There is also distribution of cell organelles and cytoplasm. Therefore, M phase is the process of final separation of already duplicated molecular units. Karyokinesis involves three, that is how this the nucleus gets divided. It divides in four again phases, that is prophase, metaphase, anaphase and telophase. And mitosis occurs during formation of body cells, therefore it is called somatic cell division. As the daughter cells resemble their mother cell genetically, it is also called duplication division or equational division. Mitosis leads to production of diploid daughter cells with identical genetic complement and helps to restore nucleo-cytoplasmic ratio. It also replaces old cells and helps to increase number of cells within the organism. That means we must be thankful to mitosis because it is helping us to replace old cells with new cells. So always fresh and young looking. Second, whenever there is an injury, the injury gets healed up, again thanks to mitosis. But where there is advantage, there is disadvantage also. When mitosis becomes uncontrollable, it leads to cancer. That means the possible thinking is whenever there is a disturbance in the ratio of cytoplasm and nucleoplasm, the cell loses its control over the division. Hence, tumors will be formed and tumors lead to cancer formation. Hence, mitosis should be always under control. Let us see in our next phase the detailed descriptions of each phase of mitosis, that is prophase, metaphase, anaphase and telophase. Until then, take care, keep drawing diagrams,